Good day. We are Group 1 and we will be discussing the development of the muscular system. Before we begin our reporting, let me first enumerate our objectives. First is to briefly go through the structure and function of the muscular system, to differentiate skeletal muscle, cardiac muscle, and smooth muscle. Third, to discuss the development of the trunk and limb, and cranial and cervical musculature, to identify the abnormalities or diseases that can arise due to abnormal muscular development, and to determine other anatomical outcomes of abnormal muscular development. For the brief introduction of the muscular system, we will first discuss its general functions, then its structure and types. The muscular system is made up of muscle tissue and is responsible for functions such as the maintenance of posture, locomotion, and control of various circulatory systems. This includes the beating of the heart and the movement of food through the digestive system. Additionally, the muscular system is closely associated with the skeletal system in facilitating movement. The muscular system includes more than 700 muscles and makes up about 40% of the total body weight. These muscles are attached to bones, blood vessels, and other internal organs of our body and are mainly composed of skeletal muscles, tissue, tendons, and nerves. Many disorders, injuries, and diseases can affect how muscles work. These conditions can cause muscle pain, muscle spasms, or muscle weakness, and more severe disorders can even lead to paralysis. Voluntary muscles are long, multinucleated cells containing sarcomeres arranged into bundles. These muscles are composed of cylindrical fibers and are usually attached to bones and the skin. They play an important role in allowing the body to move by contracting and relaxing, and their actions are mainly under the control of the somatosensory nervous system, while the involuntary muscles are striated and branched in the case of cardiac muscle. The actions of involuntary muscles are mainly controlled by the autonomic nervous system in the body. These involuntary muscles include smooth muscles and cardiac muscles. Every movement in our body is the result of muscle contraction and are found in every organ, including the blood vessels, heart, and digestive organs. In these organs, muscles function by transferring substances throughout the body. There are three types of muscle and are mainly classified based on their movements and structures. Skeletal muscles are a form of striated muscle tissues and is mainly found attached to bones by the tendons, while smooth muscles are found inside of organs like the blood vessels, stomach, and intestines. These muscles help in the movement of substances through the organs. And cardiac muscles are found only in the heart and are mainly responsible for pumping blood throughout the body.
Now that we have reviewed the muscular system and the different muscle types, we can now discuss the cell and tissue development of the muscular system. As with most cells and tissue types, muscles begin their developmental journey through the induction of their precursor cells or the stem cells that would later give rise to muscles. Though there are a few exceptions, muscle tissue generally originates from the mesoderm. Specifically, the paraxial mesoderm or the somites would differentiate to the myotome, which will then give rise to the skeletal muscles of the body. On the other hand, the splanchnic mesoderm associated with the viscera and the heart would then give rise to the smooth muscle and the cardiac muscle components of the associated organs. For the muscle precursors in the mesoderm to develop into muscle, they are usually induced to differentiate by the surrounding tissues through molecular signals. For the induction of myotome formation as shown in the figure, we can see inducing signals such as WNT, SHH, Notch, and BMP, among others, being released by nearby structures like the neural tube, notochord, and lateral mesoderm. These signals causes changes in the precursor cells, usually leading to the expression of muscle-specific or muscle-related genes. Once induced, the precursor cells undergo determination to become muscle cells, or in other words, the precursors gradually become more committed to develop as muscle cells. Determination occurs gradually through the stepwise expression of different muscle-related or muscle-specific genes, which are usually transcription factors, each of which having a different effect on the cell's metabolism, behavior, and their commitment to a pathway. For skeletal muscles, expression of these genes leads to the morphogenetic movements and migrations of precursor cells to their final locations, myoblast proliferation or the division of the myoblast cells, and lastly, the muscle differentiation. Based on the figure below, we can see that when a pluripotent cell is induced to express mesogenin 1, it differentiates to become a pre-somatic mesodermal cell, or a precursor to the somites. Further induction of these cells to express PAX3 and PAX7 genes causes the cell to become a myogenic progenitor cell, and when these are further induced to express MYF5 and MyoD, they differentiate to become a myoblast cell, the immature form of a muscle. At this point, the myoblast is now fully committed to become skeletal muscle. The final expression of MRF4 and myogenin, among others, eventually leads to the terminal differentiation of the myoblast, preventing it from further proliferation and leading to the formation of the myotube. As for the other muscle tissue types, slightly different signaling molecules are involved, but the general process of induction, determination, and differentiation are more or less the same. Now let's move on to the development of the different muscle tissue types. For skeletal muscles, as discussed earlier, progenitor cells are induced to become myoblasts, the immature muscle cells. These are spindle-shaped mononuclear cells which could continue dividing to form more myoblasts, or it could differentiate into the myotubes. For myoblasts to become myotubes, first they align themselves to each other, then fuse together as one long cell. They become syncytial, sharing a cytoplasm containing the multiple centrally located nuclei of the individual myoblasts. This process is neatly summarized by the top diagram. These myotubes will then continue its differentiation, synthesizing contractile filaments on top of other cellular changes until it eventually becomes a mature skeletal muscle fiber, a long cylindrical multinucleate cell filled with contractile filaments commonly seen in adult histological sections. As the myotubes mature, the then central nuclei eventually move to the periphery as seen in the comparison of the lower images. The left image is a skeletal muscle cross-section showing myotubes with centrally located nuclei from a 13-week-old fetus, while the one on the right shows the same tissue featuring mature muscle fibers with peripherally located nuclei from a newborn baby. For the cardiac muscles, the development starts with cardiac myoblasts, which are very similar to the skeletal muscle myoblasts. However, the differences is that these are from the cardiac primordia, and interestingly, they have the ability to continue dividing even after differentiation. This is very crucial since this allows the heart to continue growing while contracting at the same time during development. However, cardiac myoblasts do not form myotubes. Instead, they only form specialized intercellular connections called intercalated discs, forming rows of cardiac myoblasts connected end to end. Eventually, these cells would mature through various cellular changes such as the accumulation of contractile proteins, becoming cardiac muscle fibers, which is composed of multiple cardiac muscle cells joined end-to-end by intercalated discs. Lastly, it is worth noting that some cardiac muscle cells develop to become nerve-like conducting cells, capable of carrying electrical signals. These cells would then become the conducting system of the heart, including the SA node or the sinoatrial node, the AV node or the atrioventricular node, the bundle of His, and the Purkinje fibers. This conducting system is crucial in maintaining the rhythm and synchronicity of heartbeats. Smooth muscle cells arise from smooth muscle precursor cells, usually localized in splanchnic mesoderm surrounding the organs that they would eventually cover. Eventually, these precursor cells would begin arranging themselves parallel to each other, making themselves appear more organized as compared to other neighboring mesenchymal cells. This is seen in the top photo showing a cross-section of human fetal intestines. Here we can notice that layer number 3 have cells parallel to each other arranged circularly in the intestine, which corresponds to the developing smooth muscle cells. Eventually, these precursor cells would mature, becoming more elongated in spindle shape, accumulating myofilaments and other cellular changes like the skeletal and cardiac muscles. These are represented in the bottom picture showing mature circular smooth muscles of the intestines.
Let us now move on to the development of body musculature. Spindle-shaped myogenic cells, or presumptive myoblasts, travel toward the region that would be inhabited by the mature muscle from a shared origin in the paraxial mesoderm. Through a fibrillar extracellular matrix, these cells shall move. The division of the common muscle mass into the primordia of distinct muscles represent the subsequent stage in the development of a muscle. Early myotubes have started to form from the myoblasts at this point in muscle development, and frequently their orientation already matches that of the mature muscle's fibers. Later on in fetal development, particular muscle fibers groups interact with sensory nerve fibers and change into the specialized intrafusal muscle fibers of muscle spindles, which are the stretch receptors of a muscle. After the neonatal period, the majority of muscles do not add appreciable amounts of new muscle fibers, but each muscle fiber grows by gaining sarcomeres at either end and by expanding its cross-sectional area. For the development of the trunk and limb musculature, recent research demonstrates that all of the muscles in the body's trunk and limbs originate in the somites. The individual cells that form the limb muscles separate from the somites' ventrolateral edges and go into the early limb bud individually. The migratory cells create significant pre-muscle masses in the flank, which quickly separate into 4 muscle masses that correspond to the 3 oblique muscles of the abdominal wall, as well as the abdominal rectus muscle. Furthermore, the myotomal area of the growing somite is where the progenitors of the intrinsic back muscles stay, rather than leaving. Let us now move on to the development of cranial and cervical musculature. It is now widely accepted that the paraxial mesoderm is the primary source of the cranial musculature and that at least part of the cells making up the extraocular musculature has crossed the precordial plate. The connective tissue in which the myogenic cells develop appears to control the morphogenesis of the cranial muscles. Lastly, the neural crest origin of the muscle-associated mesenchyme, in the case of the muscles of the face and ventral neck, indicates that the cranial neural crest is likely to have extensive morphogenetic information.
Abnormal development of muscular system. Muscle growth mainly depends on myostatin and growth differentiation factor 11. Myostatin is a control factor that affects both the number of myofibril during development and postnatal muscle growth. Without this, an excessive amount of muscle mass is developed, thus, abnormal development of the muscle occurs. Muscular abnormalities are classified whether primary, secondary, genetic, or neuromuscular. Some causes of muscle abnormalities include age, genetic disorder, heredity, injury, and medication. Examples of muscle disorders include polymyositis and dermatomyositis categorized as inflammatory myopathies and Duchenne muscular dystrophy, a genetic condition that results to muscle weakness. We also have myasthenia gravis, an autoimmune disorder that affects the neuromuscular junction. It is a muscle weakness involving respiratory muscles. Amyotrophic lateral sclerosis is a condition caused by muscle injury, whether traumatic or non-traumatic. And rhabdomyolysis, also known as Lou Gehrig's disease, it is a fatal neurological disorder under motor neuron diseases, and it is a progressive muscle atrophy and weakness.
The interactions between muscles and tendons are vital for the development of the organism and its musculoskeletal function. Otherwise, the voluntary and involuntary motions of the organism will be compromised. Schuster, Gosler et al. Proposed that premature and excessive differentiation associated with the Notch ligand delta 1, which controls early myoblastic differentiation and maintenance of myogenic progenitor cells, results in the depletion of progenitor cells, cessation of muscle growth, and ultimately muscle hypertrophy. As muscle fiber formation is completed near birth, the repercussions of abnormal development are often manifested after birth or hatching. Skeletal muscle weakness, atrophy, and myotonia with progeroid features are common symptoms of myotonic dystrophy 1 and 2, in which inherited genes cause abnormalities during skeletal muscle myogenesis in the distal and proximal muscles respectively. Over time, physical strength and independence are lost. Partial or complete muscle absence is common and often not damaging, such as the partial or complete absence of the palmaris longus or quadratus femoris muscles. However, the pollen sequence, which occurs in 1 per 20,000 individuals, is a graver defect, indicated by the absence of the pectoralis minor and the partial loss of the pectoralis major. The nipple and the areola are absent or displaced, and fused digits or short digits are also observed in patients. The disfiguring nature of the defects may impact breast development in females. Prune belly syndrome is characterized by the partial or complete absence of abdominal musculature, in which organs become visible and easily palpated because of a very thin abdominal wall. These defects cause fluid accumulation, enlarging the abdomen and leading to abdominal muscle atrophy. Going back to our objectives, the muscular system holistically provides strength and allows movement, and is divided into three types of varying structures. Skeletal muscles originate from the brachial mesoderm, while the cardiac and smooth muscles originate from the splanchnic mesoderm through muscle-specific gene expression and the gradual commitment of precursor cells. During development, myoblasts fuse to form myotubes that give rise to skeletal muscle fibers. Inversely, cardiac myoblasts do not fuse. Instead, they form intercellular connections that become become intercalated discs, joining the cardiac muscle fibers. Smooth muscle cells also do not form myotubes and require no margation during development. In the musculature development, trunk and limb muscles all arise from the somites, while cranial muscles primarily arise from the brachial mesoderm. Without myostatin and GDF11, which primarily influences the number of myofibrils, muscular abnormalities arise. These are divided into primary, secondary, genetic, and myopathies. Anatomical outcomes such as skeletal muscle atrophy are other repercussions of the abnormal development of the muscular system. That concludes our report. Here are our references. Thank you.